Hmm. Welcome. We did, or most of you did, some stuff on plants, and that is what we are going to be focusing on today. I will begin by answering the questions that have been asked so far. So, from booklet one, I've been asked one, two, three, six. Um, that's fine. Yeah. Most people, depending on which city you're in, most people did the test the weekend before. If you haven't done it yet, it's fine. Tests can get in the way. But let me get on with the questions that have been asked. So one, two, three, eight, and six. So one, two, three, six, and eight. Okay. Um, let's have a look. Question one says, the drawing below shows Rebecca pulling a turnip out of the ground. So these things grow. um, These things grow in the ground, and what we do to get them out is pull them up out of the ground in that direction. So the answer to this question is A, because that's the direction in which Rebecca's hand will have to pull this to get it out of the ground. I will get to all of the questions, don't worry. Question B. Root maggots eating a turnip. Damaged roots don't grow very well. Damaged roots cannot take up. So the job of the roots is to take up two things from the soil. They are water and minerals. And I think that is the answer to one of the later question as well. This shows a food chain. Now, you may not have done stuff on food chains, but in a food chain, the arrow means eaten by. So what we're saying here is that a turnip gets eaten by some maggots, and then these maggots get eaten by these beetles. Now, a predator is something that eats another animal. That's what we call a predator. So in this case, when it says which word describes a rove beetle, the answer is predator. Let me explain the other answers. A producer is the very first thing in a food chain. It's always a plant. So that would be the turnip in this case. A herbivore is something that eats only plants. So the maggots would be a herbivore. And prey is something that's eaten by a predator. So maggots are also prey in this specific example. Right, so that was that one. I've got a few to do from booklet one first. I'll get through those, then I'll move on to the next one. Um, what part of a plant makes food? The best answer is probably the leaves. You could say chloroplasts, probably is an answer to that. And then what will the turnip plant use stored food for? You could probably say that it will use it to grow or for energy. Okay. Um, Yep, I'm going to do all the questions you're asking, don't worry. I'll get to them. One, two, three, eight, and six. Right, two. Richard wanted to find out the best conditions for growing lettuce plants. He took 4 trays and planted 8 lettuce plants in each. The results are below. Right, so what we did with all these plants, we used different conditions, as you can see here, depending different amount of light, different amount of air, different amount of moisture in the soil. How many days did Richard's investigation last? Well, it tells you here because he did this for 7 days. So that was that. Look at the table. Which variable did Richard change in his investigation? So he changed the light level. He changed the air temperature. And he also changed the soil moisture. So I think the answer here is trying to figure out why it would not be any of the others. The answer is air temperature and soil moisture. Oh, because he didn't change the light level. The light level was always medium. So we didn't change that, did we? It was always medium. That's why that's the answer to that question. Um, okay, I've got all your questions, don't worry. I'm getting to them today. Richard said, lettuce plants grow better at medium light levels than other light levels. Why are we not able to make this conclusion? Well, we don't know if different amounts of light make a difference here because we never changed the amount of light. It stayed the same. So we can't judge what happened if this never changes. If we had done one with like loads of light and then had a look at how many plants died, then we'd have an idea. But we didn't change the light level. We did not change the amount of light. So something like that. He did not change the amount of light. So I'm using a really bad pen today. Right, that was that. And then D, the table below shows the number of lettuce plants alive at the end of day 1 and day 7 of the investigation. For each tray, A, B, C, and D, suggest the number of plants that were alive on day 4. Okay. Well, I would say that if there were 8 plants alive at day 1 and 8 plants alive at day 7, there were probably 8 plants alive at day 4. It feels like a reasonable suggestion. If there were 8 alive on day 1 and 6 on day 7, we could probably say there might have been 7 on day 4 because one of them might have died on the way. With these ones, there were 8 on day 1 and 2 on day 7, so maybe 4, something like that, 4 or 5. There's a few answers you could say. And if there were 4 on day 1 and none on day 7, then anywhere between 4 and 0, like 2 or 3 would be fine, or even 0 would be fine there. 1, 2, 3, 6, and 8. Okay, 3. Nadine mixed grass seeds with sand. She put the mixture into 3 mesh bags to make 3 model heads. And so 2 of the bags in water, which show, and this will happen after 1 week. Now, the 2 that are in water are the ones that are growing grass on the head. And the one that isn't is the one that we didn't do that. So when it says which 2 model heads did Nadine soak in water, it's got to be A and C because they've got grass growing. And as you know, plants need water to grow. Nadine watered both of these models for 2 weeks. He watered one more often than the other. How would the model that was watered more often look different? Well, probably it will have more grass will have grown, or it would have longer grass, more grass, longer grass, something like that. Oops. So if anybody just arrived, I'm just going through a few questions in booklet 1 and I will move on to the other ones. We put one of the water models near a window. Why did the grass grow towards the window? It's because plants grow towards the light. That's what they do. They always grow towards where the light is coming from. That's why it's sort of curled towards the window. Okay, and then C on here. Grass plants have root hairs. Which diagram shows root hair cell? This is slightly unrelated, but you may have done it a few months ago when we did cells. But root cells, root hair cells, always look like this. They have this weird sort of shape. And then, as I said earlier, roots take in water and minerals from the soil. There you go. 1, 2, 3, 6, and 8. Right, bindweed is a plant that grows tightly around other plants. The drawing shows bindweed growing around a rosebush. So this is the rosebush, the main thing you can see. This weed here is sort of growing around it. That's a different plant that's growing sort of next to it, but it's using it to go upwards. They grow as high as possible so that they can get as much. Well, why is it growing upwards? It's trying to get as much light as possible. That's what it's trying to do. And it grows around the rosebush because the rosebush provides support. It helps it kind of, it's like using it as a ladder to go upwards. A gardener cut through the stem of the bindweed at X. Right, if we cut with some scissors here, this will die. But why will it die? It's because it won't be able to get any water. You've sort of cut off the supply of water from the soil because you've cut it there. So it'll run out of water and then it will die. Because we don't like weeds. They add fertilizer. Fertilizer provide minerals. What is the answer to that question? And then the root hair is the same one again. So we need to do that. And then the last one on here was 8. Although I think someone asked 7 as well. So I want to do that as well. 7. This is the equation of photosynthesis. I would highly recommend you remember this, particularly for the next verbal test that you do. Plants need water and carbon dioxide for this to happen. And they make glucose and oxygen. Jonathan studied a blackberry plant growing in a shady place and a blackberry plant growing in a sunny place. He found that the plant in the shady place had bigger leaves. Why is it an advantage for plants in the shade to have leaves with large surface area? So leaves catch the sunlight. So if you're growing where there's not much light, you want to have as big a leaf as possible to try and catch the little light that there might be. So they are able to absorb more light or get more light. Something about more light would be the answer to that question. Um, and then green leaf, which part of a leaf makes the leaf green? Which part of the leaf cells make it green? Chloroplasts is the answer, or you could say chlorophyll, which is the green stuff inside the chloroplasts. Um, this shows a cell from a leaf. Match the names. So, oh God, right, the cell wall is the outer layer of the cell. So that must be D, I want to say. Yeah. Cytoplasm is the liquid inside, so it's usually pointing at nothing like F is doing. That's F. The nucleus will be this thing inside here. That's A. And the vacuole is the big bit in the middle, which is the C. And which two of the labeled parts are also present in an animal cell? So an animal cell also has a nucleus, so you can say A. And it also has a cell membrane, so not the cell wall, but the cell membrane, which is E, I guess, is pointing just to the inner layer, inner wall. Okay, and then are there any more from booklet one? No, there aren't. Great. So question 8 is the last one from here. Alex poured some pond water into 3 beakers. She then put water weed into each beaker. She put the beaker in different places, right? So we've got some in normal daylight, some in the dark, some in bright light. In which beaker did the water weed grow best? Well, I would have thought in bright light. The brighter the light, the better a plant grows. So it'll be that. The water weed in the box changed from dark green to pale yellow. Why did this happen? Well, because it didn't have any light. That's the point. Inside the box, there's no light. So it starts to die and change color and weird stuff starts to happen to plants. Um, in the school pond, there were lots of water lilies with large leaves covering the surface. There were not many plants growing below the surface. Why? So if there's a pond, this is a pond, okay? And there's loads of stuff growing on the top of the pond, and stuff tries to grow down below. The problem is that if there's any light, what all the stuff at the top is going to take all of the light, and there won't be any light getting down to the bottom. So the reason there's not many plants growing at the bottom is because less light reached the lower plants. In another experiment, Alex put similar pieces of water weed into 2 more beakers of pond water. She added fertilizer to one of them. Remember, fertilizer is a good thing. It adds minerals and they grow better. And she kept them both by a window. So this one contains the fertilizer, this one doesn't. Suggest the rule of the experiment. So I would guess that the weed in this beaker will grow better than the other one. Okay, it will grow bigger or better or faster. So this one will grow faster because it's got the minerals from the fertilizer. I'm just doing it in order that I'm doing the question in order that has been asked. So I'm just getting through the last question on this one, then I will just do it in order that the question's been asked. What do fertilizers contain? Minerals. Okay, and that is the end of that one. Right, let me move on. So somebody asked me 4C on booklet 2. Let's have a look what 4C says. 3 ways in which plants use glucose. Right, this is a very important question. And again, I would highly recommend that you try and remember this for when you next do a test, because I can tell you. So the biggest reason that plants use glucose is for a process called respiration, which releases energy for the plant. The other answer is a little bit harder. Okay, but the let me think about them. There's a few answers here. The best answer is they use the glucose to store it, but you have to say they store it as this stuff called starch. Um, so what plants do is they can't always make glucose because it's not always sunny, right? At night, for example, right now, plants are not doing photosynthesis because they can't make, there's no light. And so they have to, when they can make it, they will sometimes store it for later, which is kind of what your body does as well, which is why you're not eating all the time. And then another answer would be to make, that's a bit harder, this cellulose. Cellulose is the stuff that cell walls are made of. and only plants have those. So that's that. Um, um, oh God, more questions. Booklet 1.5a. Right. I will maybe go back to booklet 1. But do try and ask these questions at the beginning if possible. Um, right, that was booklet 2's 4C. Um, 2, booklet 2, booklet 2, booklet 2. There's nothing else for booklet 2. Fabulous. Um, I'll just go back to 5A on 1 and see what that was. Yeah, I mean, I'm okay. Give the name of part A. I'll tell you that part A is a chloroplast, but then I'm going to let you try and find out what the function is of a chloroplast, because it's basically what we did. The name of part E looks like the nucleus, and once again, I'm going to let you try and work out what the function is of the nucleus. That's that booklet. All right, I want to move on to booklet 3. I believe now would be a good time. So 1B. Oh, God. 1B. Oof. Describe the process of transpiration. Right. Firstly, let me just explain what transpiration is. It's quite a difficult thing to understand. Here is a plant. Here are the leaves. This plant. Here are the roots of the plant under the ground. Okay. It rains and then there's water. The water gets taken up by the roots and it's sent up to these leaves. So these leaves and the whole plant is actually full of water because it takes it up and keeps it there. But the leaves contain these tiny little holes called stomata. And whenever they open, and they open whenever the plant needs to let gases in and out for photosynthesis, whenever they open, a little bit of water gets lost. It evaporates and gets lost from the leaf. But the leaf doesn't want to lose it. So what it does now is takes up more water from the soil, and it keeps taking up more, and it keeps losing some, and it keeps taking up more, and keeps losing some. And so there's like this constant stream of water going from the bottom up through the plant. That's what transpiration is. Now, to answer this question, um, where are we? 1B. We would say that um, transpiration is when water is lost from the leaves. That's the first thing. Okay. Through the stomata. You want to try and use these key words. That's probably enough, actually, but I'll just say a little bit more. More water is then taken up and moves through the plant. It's like, I can't show you it, but it's constantly sort of moving because it's being lost here and then it's more's being taken up here. Okay, fine. 2I. Is that 2BI? I want to assume it is. Yeah, well, it's not A. Um, okay. So this question here, I'm not going to write it down. I'm going to say it. Describe the relationship between the mean number of stomata per millimeter squared and carbon dioxide concentration. So this graph is showing you the relationship between the number of stomata and the amount of carbon dioxide. It shows you that when the carbon dioxide is low, there are lots of stomata. You see how that's really high up here when there's very little carbon dioxide. Then, as the carbon dioxide increases, the number of stomata falls very quickly. That's why it's such a steep drop. And then it levels out at around whatever number, at around 2, roughly, and stays there. So you want to explain that it's very high. The number of stomata is very high when the carbon dioxide concentration is low. As the carbon dioxide concentration increases, the number of stomata fall quickly and then level off. Something like that would it be sufficient to say. Right. 4, 5C, 5E. Right. Question 4. That is the equation of photosynthesis. In winter, when days are shorter, glass houses are heated to keep the enzyme reaction in plants at optimum rates. What else should a grower do to make sure the plants are at the optimum rate? To give a reason for your answer. So, apart from heating, the other thing that we need to do is keep the light intensity high, so provide more light, because if you provide more light, there is more photosynthesis. And also, there are three main things we need for photosynthesis to happen fast. We need lots of light, lots of warmth, and lots of CO2. They've already mentioned that they're heated, so what else can we do? We can have lots of light and lots of carbon dioxide. That will get your plants to grow quickly, as quickly as they can. Bum, 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 1B, 2I, 4, 5C, 5E. Oh, God. Right. A student investigated photosynthesis using pondweed. They set up the apparatus, they counted some bubbles to see it photosynthesizing. The independent variable was, what did they do? Bulbs of different power. So they changed the power output of the bulb. That's what an independent variable is. You wouldn't necessarily know that yet. That's okay. Two ways they could be improved so the results would be more valid. What question is this again? 5C. So generally, an easy way to improve any investigation is to repeat the experiment and calculate a mean. It's much better than just doing an experiment once. Do it lots of times and work out an average of your results. Also, you might want to, if you're measuring how light affects this, then what you want to do is keep the temperature constant all the time, because otherwise that will affect the bubbles. There are the results. You can do that. And then I'm not going to do 5E because it's drawing a graph based on the information that you have earlier. 6C is the last one on here. Three ways in which plants use glucose. I've already answered that earlier. You'll have to watch it back because it was in the previous booklet. The three ways in which plants use glucose. They use it for respiration, they use it for storage, and they use it to make cellulose. These are answers to that question. It was the same as what we did earlier. Right. Let me then go back and answer a couple of the remaining questions. I won't answer more questions, I don't think, from here, but 6A, let me decide if I want to do 6A booklet 2. Yes, I do. The layer of cells lining the upper surface and lower surface. So hopefully, I might be wrong, but hopefully in your notes, you will have had something that looked like this, like that, which is pretty much the answer to this question. Because it says the layer of cells lining the upper and lower surface of the leaf, that's the top and the bottom here, are called epidermal tissue. So epidermis is the answer. And then the part of the leaf where most photosynthesis occurs is this little bit here, where there's loads of chloroplast. That's called palisade mesophyll, which is there. So that's the answer there. Water is transported to the leaf in the, and you may have come across this, but these things called xylem are a bit like the blood, but in a plant. They transport the water around. So that is 6A done. And then the last one is 4D and 5A from booklet 1. Did I not do those? Okay, right. Let me do a proper explanation of this. So I'm going to write down the equations for photosynthesis and respiration. Respiration is something that all living things do to make energy. They use glucose and oxygen, they use those two, and produce carbon dioxide and water. It's what you're doing right now. And they also then get some energy out of it. It releases energy, this process. All living things do this, okay? All. Photosynthesis, though, is only done by plants because plants don't eat food the way we do, so they have to make their own glucose. They do that by doing this backwards, basically. So they use carbon dioxide and water, and they make glucose and oxygen using light. So now that we have got all of that written down, this question should become easy. The statement says carbon dioxide is produced. Is that true for photosynthesis or respiration or both? Well, produced is the other side of this arrow. Carbon dioxide is produced here in respiration. Tick. Light is needed. Light is only needed in photosynthesis. Tick. It occurs in plants and animals. Only respiration happens in both plants and animals. Animals don't do photosynthesis. You don't do photosynthesis. Oxygen is produced, again, the other side of the arrow. If there's oxygen, then that's the one we want, which is photosynthesis. And that becomes the answer to that question. Do stomata allow light to enter the leaf? No. Stomata are only there for gases to come in and out. So carbon dioxide, water, oxygen, and water vapor. And 4D and 5A. I'm not doing 5A, I've done that. 1C, part 2, part 3. Let me have a look at that. 1C. There isn't a one. Oh, booklet 3, 1C, There isn't one. Do you mean on A? I don't know what you mean. 1C, There isn't a question that you've asked. Right, is there anything else? Anything else that anybody want to ask me? If there was one thing that you guys would want to remember, oh, something has come in. 6E booklets too. I thought I'd done that. Can I not do that? Function of the stomata to let carbon dioxide into the leaf. As I said, it's about the gases. So to let carbon dioxide into the leaf. And 1B, I don't know this. 1B, I still don't know what question you mean. 1B, and they got loads of I's. 1B, I. We've only got 1B in this booklet. 1B, I meant. This is booklet 3. Oh, got it again. You don't have. Oh, interesting. Is that because you have a different booklet? It's interesting. Hmm. Is it the plants one? It is the one the plants. What does it say at the front of it? Just like that's what you need to ask me. You need to just check. What does it say on the front of your booklet? Just type that out. CO2 comes in because if we just go back to wherever I wrote the equation for yeah, keep going. Plants and photosynthesis. Keep write all of it. Say all of it. The bit after it as well, booklet number. So I don't know where I wrote that down, that really useful stuff I just wrote down. So plants need to take in carbon dioxide because they need it for photosynthesis. So the photosynthesis is the opposite of what you and I are doing. They are taking in CO2 and then they're releasing oxygen. So that's why it goes both ways, but it does come in. Yeah, mine seems to be missing it, so I don't know what I can do. Let me see if I could be anywhere else. Shouldn't be. I don't understand how it can be missing because we've got no other version of it. Yeah, mine just that's the first page of my booklet 3. It's fine if you can't do it, it's all right. Right. Um 7A booklet 2. I have to warn you about what you're doing here. 7A. I'm not doing 7A. Well, okay, I'll do 7A. So God. So this carbon dioxide is coming in to the leaves here. It comes in for the process of photosynthesis. But then remember, plants also do this thing called respiration. Respiration, just like you and I do, releases carbon dioxide. So plants are weirdly doing everything. They're taking it in and they're releasing it, both gases. But the process that releases CO2 is called respiration. So it does take a little bit of confusion to get your head around all of that. Um, but that's that. The answer to this question, in case you wish to know, is dry. Wilt is what happens to a plant when it doesn't have enough water, and that tends to happen in dry, hot weather. Yeah. Year 11. Wonderful. Right. Any other questions? I don't think there seem to be. In which case, I will leave. Oh, yeah, I was going to say something, and then you got distracted. I was going to say that for you guys in Key Stage 3, please make sure that you have learnt the equation for photosynthesis, which is here. At least the word equation. If you can get to the symbols, wonderful, but at least do learn the word equation. And then the other thing, try to remember that there's three things that make photosynthesis happen faster. The light intensity, if it's brighter, then it happens faster. The warmer it is up to a point, the faster it happens, and the amount of carbon dioxide there is. Those three things are what farmers are always trying to increase to try and grow their stuff quickly. 1 to 7 in KS3. I don't know what booklet number you mean. And the answer probably is no, depending on the booklet number. I also don't know who you are, which I'm not a fan of. Year 7, booklet 1. Yes, so it's be booklet 1. I have done all of those, so you'll just have to watch it back. I have literally done all of questions 1 to 7 at the beginning of this. So it is all available to you, don't worry. You will be able to get access to everything I did at the beginning of the video when you get it on YouTube. You will find me having done questions 1 to 7, pretty much all of them. Maybe with a couple of exceptions, but I'm also not I wouldn't have done all of questions 1 to 7 anyway. Um, you need to have an account for the website. Um, it is included. You have it. You just need to, if you don't know how to access it, you need to just tell us and we will set you up with an email address. But send us a message on our WhatsApp through your parents, and I will make sure that you have the login details that you need for the website. Okay, fine. Still don't know who you are. You can still say your name in the question, but it's the yeah, as I say, 1 to 7 you will have access to. Right, I am not going to do any more. Again, for those of you that are asking about the website thing, I really, really recommend it. Possibly more so for like years 8 and 9. For 7, it might be a little bit advanced, but um, the videos there recap all of the content that you probably need. Um, but you just need to ask us for the login because even though we have set it up for you, your parents sometimes don't share that information um, with you. So that's on them a little bit. So I am going to stop now and then I'll return at 8 p.m. for anybody who might turn up that wants some answers from the Year 10 homework booklet. Right. Hello, if anybody is still here. If you have any questions on the Year 10 homework on plants or the Year 11 homework relating to the biology required practicals, it's quite a complicated booklet, this one, um, then go ahead and we can focus on that for now. Um, 3 in the back of the Year 10 booklet. Not received your biology notes. I'm sure they're on the way. 7 CDG part Year 10. Yep, fine. I'll get going with that first. So, um, yep, 2I Year 10. Sure, sure. Um, right. Do it. I'll try to do it in order. 2I. This one. So, there are two types of cell that transport things around a plant. They are these two, phloem and xylem. You need to know the difference. Xylem transports water from the roots up to the leaves, and phloem transports sugar all over the plant from the leaves where it's made in photosynthesis, and then it goes all over to be used for storage or for respiration and so on. And so, I mean, the answer to that question is xylem, then, isn't it? And then phloem is the next one. The movement of water is called the transpiration stream, which we might come on to with a different question in a moment. Um, 7 seems to be popular. Oh, 3 in the back of the agenda, 7G, right, 7. I'll do 7 and I'll do the back of the booklet. Right. Okay. 2I as well seems to have been asked. How to do that one? No, sorry, I did that. Okay. Right. These things, aphids, are small insects that carry pathogens. An aphid feeds by inserting its sharp mouth to the stem of the plant. After feeding, it contains a high concentration of sugar. Which part of the plant was it feeding from? As I just said, there are two types of cell that transport stuff around a plant. Xylem transports water, and phloem transports sugars. So what it must be feeding from is the phloem. What is the process that transports it around the plant? That is called translocation. So translocation is the movement of sugar around a plant. Transpiration is the movement of water up the plant. Really. Um, don't worry, I will get to any questions that anybody has. Just make sure you remember when you ask a question, just make sure you confirm that you are asking about the Year 10 booklet. Um, I'll just do all the questions there. Don't worry. Plants infected with aphids have stunted growth. Explain one way the removal of sugars from the plant causes stunted growth. Well, the sugar is mainly partly used for respiration. So if we are removing sugar, there's going to be less sugar for respiration. And what does respiration do? It releases energy. So if we're getting less respiration, less energy will be released. And that energy is partly used to help the plant grow. And so by removing the sugar, we're reducing the respiration, we're reducing the energy, we're reducing the growth. That's how we go about explaining that. Most aphids do not have wings when they hatch. After some aphids hatch which have wings and can fly. What is the advantage of being able to fly? Well, I mean, what do we know about this thing? All we know is what the question has told us. They like to go up to a stem and feed from the stem. So I'm guessing that the wings allow it to fly to other plants or to another part of the plant and get more food. There are other things you can say, but that seems to be the most obvious thing based on the question. The leaves of some plants release oils onto their surface. How the production of oil may protect the plant from aphids? I guess, again, what is the aphid trying to do? It's trying to feed off the stem. So it could be that the oil harms the aphid in some way. It could be that it makes it, if it's oil, it's slippery, right? So maybe it can't then grasp onto the stem. So you could say a couple of things. You could say that it harms the aphid or kills the aphid, or you could say that it makes it slippery so it slides off, so it can't feed off, and that protects the plant. This shows part of a rose plant. Give one adaptation that helps the rose plant defend itself. Again, in this entire question, we have been talking about how the this creature goes to the stem and feeds off it and takes the sugar away. What do rose plants have? They have these thorns, which presumably make it harder for something to go there, right? They're sharp. You've probably experienced them. Um, and then figure three shows a plant of a garden containing rose plants. There's the direction of the wind, right? That's a rose plant. Plant A has a fungal disease. Okay. Um, which plant is the fungus likely to spread to first? Well, if it's A is the one that's infected and the wind is going this way, I'd have thought C might be next, just geographically. Um, and it's the closest to A, essentially. That's the reason why it's in the direction of the wind. Suggests one way the gardener could reduce the spread of rose black spot to the other plants in the garden. I suppose he could remove um, A, right? I mean, one thing you could do if you want to prevent this disease from spreading is get rid of this plant A. You could say that. You could say something like use a fungicide, which helps to get rid of a fungus. I think the easiest answer is. Or you could even spread them out a little bit more because they're really close together. So if one gets it, something else will get it. So that feels like a sensible answer to that question. And that is all of that. And then that was all of seven. And then nine as well was asked. C part two. Probably have to do an earlier part of it to make sense of it. Oh, all right. So this was that. Okay, so this has gone into the other part. Okay. Um, three at the back. Yep, yep, yep. Right. I think the back, I might try and do a few of these because the back is generally quite tricky. So let's have a look at all of it. Um, where does it start? Here. Describe how and where oxygen is produced in photosynthesis. Firstly, let's just talk about that. So, firstly, the place where photosynthesis happens is chloroplasts. Okay, and what do? They use the light energy and water and carbon dioxide. Remember, if you know the equation of photosynthesis, you should be able to answer this question. That's really what it's asking you. So water, light, and carbon dioxide are used to make, well, glucose and oxygen, but that is where the oxygen comes from. Explain why the amount of oxygen gas given off is not a true measure of the rate of photosynthesis. It's quite a tough question, that. So remember, plants do both photosynthesis and respiration, which is confusing because in photosynthesis, they use carbon dioxide and water to make, I've just mixed up these things, glucose and oxygen. But then in respiration, they use glucose and oxygen to release energy. So some of the oxygen that's being made here gets used straight away over here. So it won't necessarily always just give off all of the oxygen. It'll keep some of it for itself. And so that's why it's not a true, just seeing how much gas is given off doesn't tell you how much photosynthesis is happening necessarily, because respiration is also occurring. Because remember, all living things respire. Um, and some of the oxygen is used up in respiration. So it's not all released. Some of it's kept. Um, right. Yeah, let's leave that. Question three at the back. Right. This looks complicated. Um, this grass is a plant that grows on exposed areas of sand dunes. That to me suggests quite a dry place, hot. Figure 19 shows this grass growing and a cross section through a leaf of marram grass. So there it is. What a great picture. Then it's got these stomata, that's the upper surface of the leaf, thick waxy cuticle, trapped air in the center, hairs on the lower surface. Explain how this is adapted to survive in the hot, windy, and dry conditions of a sand dune. Right. So, um, there's a few different things that we can say. What are the easiest answers? Right. What do you know about stomata? Stomata is these little holes that allow carbon dioxide in, but crucially, wherever there is stomata, there is transpiration happening as well. Transpiration is the loss of water from the leaves. if you are growing in these hot places, hot, windy, dry places, you don't want to lose that much water. It's one of the reasons why cactuses don't have any leaves. Um, and so the stomata are only sort of on the sort of lower surface. They're not on the outer upper surface of the leaf. And so that's one way of preventing water being lost. So you can say that there's no stomata on the upper surface of the leaf, or you can just say less stomata to prevent water loss. Okay, anything else we can say? Um, yeah, the other thing about this is quite hard, this one, but this thick, waxy cuticle, because it's thick, it prevents water from escaping easily. It's like having a thick cell wall, if that makes sense. You know what I mean? Like you have the a thick wall, reduces diffusion. Um, so thin walls are good for diffusion, thick walls are bad. So you can mention that the thick, waxy cuticle also helps to reduce water loss by evaporation. Or I'm probably going to put by diffusion. Okay. Um, Someone has commented. Well, it's not that it's not well. So the question is, if there's no stomata, how is it good? It's not that it's good or bad. It's just that one of the problems with stomata is that they are one of the places. It is the only place where a plant loses water. So yes, they're needed for photosynthesis to happen. Carbon dioxide has to come in. But if there's lots of them, it will lose a lot of water as well. So in a hot, dry place, you'd want to have fewer stomata. So that is just an adaptation of plants that live in hot, dry places. They tend to have fewer stomata because they need to keep the water in. What else can we say? The other answers here, they say that see, you can't tell from this picture, but one of the answers is that grass is flexible, so it does not break in the windy conditions. Okay, fine. It's hard to tell that from the picture. Um, but again, you can't see the roots here either, so it's hard to answer this because you can't. The picture's not very good. Um, what else? I'll do one more point. Um, yes, I'm going to put the point that the mark scheme says, which is again, it's hard to tell because the picture's not very good, but windiness is means that the plants can break quite easily. But if the grass is not too like hard and thick, it doesn't break as easily. So you can mention the fact that the grass is flexible, so it does not break in windy conditions. And that should be enough for most of the marks because you get two roughly for each point if you deal with that. Okay. The second Q1, I don't understand. Oh, this one here. Um, Variegated leaf. Which diagram shows the correct starch tests result? Well, this is the test. This is the test for starch. Iodine solution turns blue, black. Um, and all the starch should be. So the answer to that question is B, because most of the photosynthesis is happening in the center of it. So it is B. So look, most of the photosynthesis happens sort of in the inner part of the leaf, so in this part over here. So where photosynthesis is happening, that's where we get our starch, essentially, where the sugar gets stored. And the test is quite a difficult one, but the test for it is iodine solution turning blue black, which you may remember from the topic of digestion. Um, IV and C. I don't know what you mean when you say these things. IV and C. Um, yeah, I need question numbers. Is it this one over here? On question 9? Okay. Question 9. The diagram shows a section through a plant leaf. Lovely. Um, Drawing on cells X are these are called guard cells, firstly, because they are the ones that control the opening and closing of the stomata. Water vapor is lost from leaves. Water loss causes a leaf to lose mass. This shows how the masses of leaves from two species, P and Q, change over several hours. So you can see that the mass is decreasing for both of these two over time because they're losing water, I guess. What was the mass of the leaf of species Q at 0 hours? That's just reading here. That looks like 2 to me. What was the difference between the mass of the leaf of species P and the mass of species Q after 5 hours? So P was on 1.9, Q was on 1.85. The difference between these two looks like 0.05 if I mentally do that. 2I and 2. Yeah, I'll do that. And then I'll just finish the question off. The leaf of species Q loses water at a faster rate than the leaf of species P. So why could that be? Um, See, the problem with this question is they were kept in the same conditions. So you can't say something like it was hotter or it was windier or something like that. These are ways in which transpiration can happen faster. The only answer probably is the difference between these two, and we talked about it earlier, is probably the number of stomata. Species Q has lost water more quickly, possibly because it has um, more stomata. You can't say any, even though an answer would normally be that maybe it was in a hotter place, so it lost more water, or it was in a brighter place, so it was photosynthesizing more. But in this case, you can't say that. You have to just talk about what the structure might be. Um, Larger stomata, interesting. Um, yeah, probably. Yes, I'm going to go with that. Yes, I would still put more, but yeah. Which weather condition caused the greatest loss of mass for both species P and C? Well, generally, transpiration, which is the, try and remember this, hopefully you'll remember this, is the way I remembered it. I was taught that transpiration, which is the loss of water from a plant, happens fastest in good clothes drying weather. Imagine if you wanted to dry some clothes outside. What kind of weather conditions would be good for that? It would need to be nice and warm and preferably windy as well, because the windier sort of conditions dry out the clothes more quickly. And so the greatest loss of mass, which is the greatest loss of water, will happen when there's wind and a high temperature. So that is the answer to that question. And in very hot conditions, the start, say close. This is to prevent wilting. Wilting is when a plant loses so much water it collapses. I'm sure you've seen them if you've ever failed to water a plant, as I have regularly. They sort of droop over. That's wilting, and that is due to not having enough water. Okay, two. Got all of that done. Yeah. Oh, no, this is more transpiration. Right. Let's do all of question two. 2I and part two. So phloem was here, as I mentioned, and transpiration stream is there. Don't need to write it all. Water vapor moves out of the plant through stomata. Is this the question that you're asking me? I hope it is. I think I've done everything else. Part B, one and two. Okay, fine. Right. Students investigated the effect of different conditions on water loss from leaves. Right. Before I go into this, water loss from leaves is transpiration. There are 3 or 4 factors that speed up the rate of transpiration. They are temperature. If it's nice and warm, you tend to get more transpiration because water evaporates more. Light intensity also affects it because the brighter it is, the more photosynthesis the plant will do. And if it's doing more photosynthesis, it will open the stomata. And as soon as it opens the stomata to let in carbon dioxide, it will lose some water. So that's another reason why light affects transpiration weirdly. It's like conversely, it does that. The other ones are wind. If it's windier, then you tend to get more transpiration. I can explain that if we come across it. And humidity as well. If the air is drier, you'll get more transpiration. If it's like humid and damp, like you know that feels like in certain countries when it's sort of moisture in the air already, then the leaves tend to lose less water because of the concentration gradient. Right. They set up some apparatus Four flasks, A, B, C, and D. They used the same size plant chute, lovely, recorded the mass of the flask and plant chute at the start of each experiment, and they left each flask and plant chute in different conditions. What they're going to do is weigh all this before and weigh it after and see which one's lost more water. Because remember, what's going to happen here is the water will be taken up and then it will go out through the leaves. And if the right conditions are there, more will go. So we'll be able to see that by weighing it. And so this was the situation. They put A at 20 degrees C without a fan. They are replicating wind using the fan. B was 20 degrees but with a fan. Then we had 35 degrees, no fan and fan. Now I can tell you before going any further that the best conditions for transpiration will be here because it's hot and you've put a fan there. Why do we use cotton wool? Right, cotton wool. Because remember, what we are measuring is the water loss that's gone through the stem and out of the leaves. That's what we're trying to measure. If we don't put that cotton wool there, then some water may just evaporate. And that's also going to be recorded in our mass, but that's not what we want to record. We want to record only the loss of water through transpiration. So the cotton wool goes there to try and reduce the water that's evaporating. Okay. Right. They had to use the same size of plant chute for a fair test. Why? Well, because we're trying to, if a plant has got more leaves, then it will have more stomata and therefore more transpiration. We are only trying to find out how the conditions affect it, not how the number of leaves affect it. So we're using the same size plant chute so that they have the same number of leaves. You can say same number of stomata if you like. Um, um, from which water evaporate. So explaining that the leaves are where the water evaporates from. So if something has more leaves, it will lose more water just by definition. Um, Table 2 shows the students' results. What mass of water was lost by the plant chute in flask D? Well, it's just, isn't it the difference between those two numbers? Yeah. You can sort of, sometimes with these questions, you can figure it out based on what else has happened in the table. So, by looking at those two numbers, the difference between those two numbers is 1.9. That's where that 1.9 has come from. Similarly with these two, that's where the 3.5 has come from. So even if you can't, interpret what's going on, you could sort of figure it out. So the answer here is 4.5. Yeah, 4.5. And as I predicted, the most water has been lost when we had the higher temperature and the fan. Um, Suggest what conclusion can be made about the effect of temperature on water loss from the plant shoot. Well, it seems as though, see, look, if we just look at the 20 degrees with a fan and 35 degrees with a fan, we lose more water. If we look at 20 degrees with no fan and 35 with no fan, we lose more water. So it seems that increasing the temperature increases the water loss. Ooh. And what conclusion can be made about the fan? Well, same thing really. Having a fan increases the water loss. Remember, the fan is just replicating wind in this situation. Right, they carried out another experiment with no fan, and they have put a plastic bag around it. Um, what mass of water would be lost from the plant chute in 2 hours, do you think, in this situation? Well, 20 degrees C, no fan, and we put a plastic bag around it. So 20 degrees C with no fan before, we lost 1.9. I think we'll lose even less now. So I reckon it's 0.3. And let me explain why. Because remember what the fan did? What the fan does is that when a leaf loses some water, it basically blows that water away so that there is still a low concentration of water there and a high concentration of water there. And diffusion or osmosis happens fastest when there's a big difference. But if it's not windy, then a lot of this water just stays there, which means there's quite a high concentration near the leaf, which means it doesn't happen as quickly because it's not as big a difference. There's not as big a concentration gradient. Um, and so I think even less would be lost, so it would be 0.3. The reason is that the plastic bag actually reduces the airflow even more. It's like the opposite of having a fan. It's just staying there now. It's not being blown away. So the plastic bag reduces the airflow. Right. Is that everything? That seems to be everything. Um, right. Someone had asked me to do some AQA triple science chemistry paper on topics that you think I need to know. And if not, can you do some moles? Um, yeah, probably. Well, let's do this. So I'm just diverting into chemistry briefly. If anybody wishes to stick around, we'll do this for about 10 minutes or so. Um, no, I don't want to go that. Don't want to go that. Okay, paper one, chemistry, AQA. That's the one I'm talking about right now. If you are doing a different exam board, you will still need to know the same stuff, just possibly in a different order. Um, right. I'm just going to go through the contents page briefly to show you paper one, what you have. So, all of this stuff hopefully is relatively comfortable. Try to make sure you know the history of the atom, which is to do with that alpha particle scattering experiment, which is really in physics, but it's also here. So you need to just be prepared for knowing that that guy called Rutherford did an experiment, and we learnt that there is a positively charged nucleus in the middle of the atom, and it's very small and there's mostly empty space. We learned that as a result of an experiment. Electronic structure 288, that stuff. Um, the stuff about this, the kind of question that I would ask someone, and it probably was in the test, which is make sure you understand and are able to explain why, as you go down group one, the elements get more reactive. And it's to do with the fact that they all have one outer shell electron. And as you go down, since they're trying to lose that one electron, it is easier to lose it as you go down the group. As the atom gets bigger and bigger and bigger and has more shells, that one electron gets further and further away and has less force of attraction and therefore is more easily lost. Make sure you're able to explain the opposite for halogens. As you go down the group seven, the elements get less reactive because it becomes harder and harder to gain the one electron that they're trying to gain. So either of those questions be ready for. The transition element, this is a triple science only thing, which is, and the main thing that you get asked about transition elements is comparing them to the group one metals, as I will prove to you now. So there you go. Comparison with alkali metals. You sometimes get asked, what's the difference between them? Why they're both metals, so why are they called different things? One's called alkali metal, one's called transition metals. Um, Firstly, transition metals are much less reactive than group one. Group one do all that fizzing and stuff when you drop them in water. They're generally much harder than group one metals. The group one metals can be cut very easily with a knife. They're very soft. They tend to have higher melting points than group one, and also they can be used as catalysts. I'm sure it says this somewhere. Yeah, yeah, there you go. They can be used as catalysts. That's enough. That's enough different points in relation to that. The next major topic is, of course, and I can't go into it now in the time that we have, but ionic and covalent bonding. Make sure you're able to answer questions about how compounds are formed ionically when one loses, when one gains electrons. And Then covalent bonding, which is the overlapping circles and the sharing business. Um, 1HI on year 11 and 4F. Yes, I'll do that in a moment. Um, and then I won't do that bit. I will possibly just come back in a moment to I'll just tell you the other topics in paper one are quantitative chemistry, this to do with electrolysis, and this to do with energy change, endothermic and exothermic reactions. That's essentially what paper one is on AQA. I will just go back and answer the one question we have had relating to this one. 1HI on year 11. Oh God, it's so where is it? Ugh. Okay. Um, okay. I mean, I have to understand this whole question now. Got to get part H. Right, let me quickly just read through this. Right, we did some of that. This was used to measure the concentration of starch present in the solution every 30 seconds. It measures the amount of light that cannot pass through a solution. That's absorbance. Absorbance against concentration of starch. Okay, the more starch there is, the more the absorbance, it seems. Um, okay, that's reading off the graph, hopefully. 1HI. The concentration of starch at 20 degrees C after 1 minute is different from the concentration of 40 degrees C after 1 minute. Isn't that just because the enzyme is working faster for that one? Yeah, so at 20 degrees C, starch is broken down less quickly or more slowly. Remember, enzymes generally work best at around 37 degrees. Um, and that's because the enzyme, it's to do with chemistry rates of reactions because the enzymes have got less energy. And that will be that. Predict the absorbance for the solution at 80 degrees C after 30 seconds. God. I need to use, um, 80 degrees C. Right. Well, 80 degrees C is very high. That means that there will be no enzyme action. The enzymes will be denatured. Um, and therefore there will be loads of starch still in the solution, like a very high amount of starch, which means that the absorbance will be about as high as it possibly can be, which, according to this, is 1.08. And that is the answer. And it's because the enzymes would have denatured at 80 degrees. That was that one. Um, so the starch doesn't get broken down at all. The key is looking at the temperature and understanding that when it's too hot, enzymes get denatured. And for F, in horticulture, wouldn't absorbance be less? Not according to the graph. See, according to the graph, oh God, I've gone too far. This is a graph of concentration of starch versus absorbance. If the enzyme, remember the enzyme, amylase, if it is, breaks down starch into sugar. So if the enzyme is working, the amount of starch is decreasing. There'll be less and less starch because it's gradually turning into something else. But if the enzyme is being denatured, then that starch will remain as starch. So the concentration of starch will therefore be high. Now, the concentration of starch is high, the absorbance is high, according to this graph. So that's why. And auxins, right. So plant hormones are used for various different things. What I would suggest, I did make some notes on this once somewhere. I'm fairly confident I'm going to find them now quickly. Uses of plant hormones. Auxins, for example, are used um, to disrupt growth patterns, which can kill weeds. They can be also used to grow plants from cuttings by growing them very, very quickly. Gibberellins can be used to stimulate seed germination, growth, and flowering, to make them germinate at times of the year they wouldn't normally. And ethene is a gas that can be used to speed up the ripening. Does that answer the question? Ethene reduce the time taken to ripen. Gibberellin promotes seed germination, and auxin can be simulate root growth in plant. I should say plant. I believe I should say plant cuttings. Let me just check that is the answer. Yes, it is the answer. Right. Okay. Since I've covered that, I will just briefly go back to the chemistry thing. I'm going to just do one question about a titration, but where will I do it? I know it says it there, but I want to do it here. Um, I will, as we approach your exams, do a lot of these kind of questions. I'm just going to do one, and that'll be it today. Okay, a titration is when we gradually add an acid to an alkali until it neutralizes. Now, you know that an acid plus an alkali gives you neutral. But that's not quite as simple as that, right? Because surely it depends on how much acid and how much alkali and how strong the acid is and how strong the alkali is. A drop of acid is not going to neutralize an ocean of alkali. Um, so what titrations try to do is work out exactly how much of one type of acid neutralizes exactly how much of another type of acid. Um, and what a student found, according to this question, was that this volume of this acid and this strength of the acid, this is a concentration, was needed to neutralize this much alkali. How many moles of sulfuric acid were needed to neutralize potassium hydroxide? Um, okay, right, let me explain how to do this. With any of these questions, and I will do lots of these in the coming months, just so you know, which is, um, I always make a little table where I have concentration, mass, volume, and then I have the acid and the alkali. Right. And there is a formula, which is concentration is mass divided by volume. Possibly you need moles. Yeah, possibly not. It might be okay. Right. And then I want to fill in this table based on what the question says. This is a volume of an acid. So 37.6 centimeters cubed volume of acid. Okay. This is a concentration of an acid, 0.5 moles per dm cubed. So I want to put in the C, acid concentration. The first thing I notice is that these units are different. So that's centimeters cubed, that's dm cubed. There are 1,000 centimeters cubed in a decimeter cubed. So to convert from here to here, you divide by 1,000, and that's what I'm going to do with this number. Divide it by 1,000, and it'll be 0.0376 dm cubed. Then this formula tells me that to work out m, you can do c times v. So if I multiply those two together, I should get this value here. And I want to do that. My calculator is around somewhere. Should do that in my head, really. 0.5 times 0.0376 will be very small. 0.0188. Now that unit of this answer is in moles because this was in moles already. So it was moles per dm cubed, which means that the mass is in moles. Right, that actually answers this first question, which is how many moles of sulfuric acid were needed to neutralize it. So we have worked that out. That's how many moles it was. That's where we got this volume and this concentration from using these numbers. Then it gives you an equation. That is the sulfuric acid, just to be clear. That is the alkali. Right. And it asks you to calculate the oh, I'm missing something. I'm missing something. Yes. It says 25 centimeters cubed of the alkali here. So the volume of the alkali is 25 centimeters cubed. But again, what I'm going to do is turn that into dm cubed, which is 0.025, because that's what I will need later. Then, this is the important part. The reason they give you this balanced equation is because, in fact, we don't even need the right-hand side, is because of these numbers at the front. There is a 1 in front of the acid, and there is a 2 in front of the alkali, which tells you that the moles are in a 1 to 2 ratio. So if I have the number of moles of an acid, I can work out the number of moles that I will need of the alkali using those numbers. If I times that by 2, it will tell me that 0.0376 moles is the moles of the alkali that we will need in this experiment. And the question finally says, calculate the concentration of the hydroxide, which is the alkali. Concentration, as that formula says, is M divided by V. I've got those here, M and V, so it'll be 0.00376. Divided by 0.025, that will work out the concentration of the alkali as 1.504 moles per TM cubed. If you can get your head around what I did there, you will definitely get a question like that, and it will basically be identical every time where you have to do that kind of calculation. Right, I'm not going to be able to do much more or any more for today. If you do have questions, try to send them prior to these sessions, and then I can sort of plan accordingly from the beginning. That'd be usually quite helpful. But next week, if you're in year 10, you will be doing, oh my God, I don't even know anymore. Actually, I have no idea what you're doing next week in chemistry. Don't know. But the year 11s are doing equilibria, reversible reactions. It's very interesting. You will be doing the other required practicals next month. We just don't quite do them all at the same time. Right, that's all.